Thank you very much for the opportunity to talk about SCOR Working Group 157, MetaZooGene. I'm delighted to be here today and to give you an update on some of our activities during the recent year. The proper title of MetaZooGene is Toward a New Global View of Marine Zooplankton Biodiversity based on DNA metabarcoding and reference DNA sequence databases, a very detailed title, and it's exactly what we're doing. I'm joined today by Todd O'Brien of NOAA Fisheries, a ZooGene member, database expert, who will address some aspects of the reference DNA sequence databases, which are a primary deliverable of MetaZooGene. And so I also would recommend to you two websites, our MetaZooGene website as well as the SCOR website, for additional information about the working group. MetaZooGene currently has a total of 23 members from 19 countries. We've done quite a good job of our global aspirations, which is consistent with the SCOR mission, of course. I'd like to mention in particular the two vice chairs, Katja Peijnenburg from Naturalis Biodiversity Center in the Netherlands, and Ksenia Kosipakova from the Russian Academy of Sciences, who are important as a leadership team and were active. We were able, luckily, to meet during February in San Diego at the Ocean Sciences meeting, and that picture is shown here. The primary focus of MetaZooGene are marine zooplankton, and we call them holozooplankton, that is, animals that drift with the ocean currents during their entire lives, although we blend a little bit into mesozooplankton, those that are just larval forms. The zooplankton consists of 15 phyla, 32 classes. The taxonomic and biodiversity complexity is a real challenge, and that, of course, is the opportunity for MetaZooGene. Our goal is integrative molecular morphological taxonomic analysis, and we're definitely focused on the global ocean. Our terms of reference include creating an open access web portal for DNA barcodes, I'll talk about that, design an optimal DNA barcoding pipeline, and best practices for DNA metabarcoding, and I'll explain those terms. First of all, what is metabarcoding? Well, you've probably heard of DNA barcoding, which is the use of a short DNA sequence which identifies species of animals and plants in some cases. For metabarcoding, this is the product of high-throughput DNA sequencing, the recent genomic revolution, which allows sequencing of environmental samples, again, for short regions, which are called barcodes, hence metabarcodes. Bioinformatic pipelines, the statistical analysis is challenging and diverse, and that, of course, is another of the goals of MetaZooGene. Now, the product of the barcoding analysis gives you detection of the diversity of zooplankton. And I've already explained that there are many phyla, many groups. The taxonomic complexity is a real challenge. This is a picture showing a survey of a zooplankton sample, which was analyzed for a short region of the 18S ribosomal RNA. It's called the Tree of Life gene, found in all living organisms on Earth. And this V9 region is quite a conserved region. It serves as a very nice barcode for groups. And you can see here many of the groups of marine zooplankton. So metabarcoding is a rapid way of looking at the diversity of zooplankton in all of their taxonomic complexity.
Hi, this is Todd O'Brien with NOAA Fisheries and Copepod, the Global Plankton Database. My role in this working group is to provide database development and product support. We have combined data from the BOLD and GenBank databases, along with soon-to-be-published working group participant data, to create a combined database of over 154,000 COI DNA sequences, representing over 9,000 species of zooplankton. In addition to adding new data, we've also added new capabilities. For example, a user can now choose only species found in a specific ocean, such as euphausids found in the North Atlantic or copepods found in the South Pacific. This is a feature not currently available in BOLD or GenBank. The MetaZooGene database and interface will be a major product of the working group and is already publicly available with additional components coming online over the next few weeks. It is important to note that the original sequence data remains in the BOLD and GenBank archive databases. The work of MetaZooGene provides an extra layer of accessibility, features, and quality controls to these already existing archive-level databases. One product of MetaZooGene, which we're referring to as the Atlas, provides tables and maps of species biogeography and their barcoding status. The Atlas can provide a listing of all species found in an ocean of choice, then tell which species have already been barcoded and which have not. This feature would allow a researcher to quickly focus their future work on those species that were not already barcoded in their region.
All of the MetaZooGene members and associate members are active researchers contributing to the forefront of investigations into DNA barcoding pipelines, metabarcoding of marine zooplankton. And I'm going to give a quick survey of some of the recent results, starting, of course, with my own lab at the University of Connecticut. In this case, we're using fisheries monitoring programs, the NOAA-NIMS Ecosystem Monitoring, or ECOMON surveys. Wonderful resource because they're regular collections throughout the entire Northeast Shelf, and the samples are analyzed for morphological taxonomic analysis by experts working microscopically. Huge effort that we can take advantage of in partnership with ECOMON. And so a recent paper of mine looked at time series diversity of zooplankton using ECOMON samples. Leocadio Blanco-Bercial of the Bermuda Institute of Ocean Sciences is another scientist focused on time series analysis. He's working at BATS, the Bermuda Atlantic Time Series Station, again taking advantage of time series collections that give him morphological taxonomic counts so that he can compare it with the metabarcoding analysis, finding really clear cycles both seasonally and in terms of community composition, which really validate the whole metabarcoding approach. Another MetaZooGene member is Bruce Deagle of the Australian Antarctic Division in Tasmania. He's taking advantage of an unequaled resource, the continuous plankton recorder surveys found throughout the Southern Ocean. Again, they are using metabarcoding for various genes, including the Barcode gene, cytochrome oxidase 1, and looking both at the zooplankton sample from the typical metabarcoding that I've described before, and also for water. And they're looking for the dissolved and suspended DNA in the water sample. This is called environmental DNA. It gives you a whole new look at the diversity of the region, not just what you caught in your net. Katja Peijnenburg, as I mentioned, a vice chair of MetaZooGene, and her colleague Debbie Wall-Palmer at the Naturalis Biodiversity Center in the Netherlands, they have what they call the Plankton team. This is a group of people who combine morphological taxonomic experts, molecular geneticists, people who are smart about bioinformatics and barcoding pipelines. They use museum collections, and they're focused on two very difficult sets of organisms. One, ketognathous amphipods, really important, really problematical in terms of their genetics, and also gastropods, which are a fascinating group because of their calcareous shells, very sensitive to ocean acidification. More results from MetaZooGene. This is Aino Hosia from the University Museum of Bergen and her colleague Louise Martell. Aino is an expert on gelatinous zooplankton, especially hydrozoans, a cnidarian group. Cnidarians, gelatinous forms are very undersampled, underknown, because they mostly disappear when they're captured in the net. They're just goo when they come up. And so what Aino has been able to do is to take lovely photographs of the living specimens, morphological identification, and DNA barcoding. It's an amazing effort, very successful. Another really comprehensive barcoding effort, which a very difficult, highly diverse, most important group in the ocean, Copepods. Tuna Falkenhaug from the Institute of Marine Research has been working on an inventory of copepods from Norwegian waters, working through a couple of programs which she calls COPCLAD and HIPCOP, in association with the Norwegian Biodiversity Information Center. She's developing an incredible resource of reference library of DNA barcodes for identified specimens. Junya Hirai of the University of Tokyo is also focused on copepods. He's also a taxonomic expert. He's working throughout the North and South Pacific Ocean, looking for biodiversity by depth of copepods, by family, and he's using a variety of genes, including another barcode region, the 28S ribosomal RNA, which he has found to be very successful in amplifying and detecting copepods across a wide diversity of taxonomic groups. Amazing in terms of its scope and its challenging group. Copepods are a very difficult group for morphological identification, and they also present particular challenges for genetics. More copepods. This is Arctic focus. Jennifer Questel works with Russ Hopcroft and Xenia Kostapakova. Xenia is a vice chair of MetaZooGene. Jen was recently a postdoc in my lab at UConn, and she has done a multi-gene analysis of copepod diversity across the Arctic, comparing CO1 and two hypervariable regions of the tree of life gene, V4 and V9, and asked the question, is there a best gene to investigate copepod diversity in the Arctic? Huge effort led by Chaolun Li of the Institute of Oceanology in China and his colleague Mingxiao Wang. This is an updated DNA barcode database in China. This is across all of the taxa found in the pelagic zone, all of the zooplankton, including many difficult forms, and so they're using multiple genes, amazing numbers, 3,700 sequences, and 350 species. So huge effort, both geographically and taxonomically, and in terms of the DNA sequence data that have been produced. MetaZooGene's mission is definitely global, and we were delighted to recruit Jenny Huggett from the Department of Environment, Forestry, and Fisheries from South Africa with an amazing team spread throughout the country with experts, morphological experts, barcoding, metabarcoding, focused on marine zooplankton in very dynamic, very commercially important regions around South Africa. Now, one of the products, of course, from MetaZooGene will be publications in the peer-reviewed literature. We're very pleased that partner with the ICES Journal of Marine Science. ICES is the International Council for the Exploration of the Sea, a good partner for SCOR and MetaZooGene. And we were delighted when they accepted our request to consider a special issue for patterns of biodiversity of marine zooplankton based on molecular analysis. We're still accepting papers. It's an open call, and it's attracting quite a bit of attention. Recent publications, recommended readings. This isn't a complete list, certainly, but there's one very nice review by Silke Lachman, Leocadio, Blanco-Bercial, and Astrid Cornel, all MetaZooGene members, microscopy to genes in biodiversity, looking at copepods in the Philosophical Transactions of the Royal Society. Katja Peijnenburg and colleagues have just published a paper in the Proceeding of the National Academy of Sciences on pteropods, evolutionary history. And I called out also a paper by Junya Hirai, a recent metabarcoding analysis of copepods in the Pacific, and +1. This is, of course, a very incomplete list, so I do encourage you to look at additional papers from the on the website. With that, I'd like to acknowledge all of the members of MetaZooGene SCOR Working Group 157. It's an amazing team, very productive, very engaged, and genuinely global. And I'd also like to acknowledge a partner program, a partner group Again, in ICES, it's the Working Group for Integrative Morphological and Molecular Taxonomy. We have shared missions and we work together very effectively. And I'd also like to acknowledge the foundations of a lot of what we do and some of the same people from the Census of Marine Life field program in marine zooplankton that was active from 2004 to 2010. Thank you very much for your attention. I appreciate the opportunity to talk about MetaZooGene SCOR Working Group 157. Thank you very much.